Welcome back, friends. This is Professor R.M. Mahendrakar from JSS Arts, Science and Commerce College, Gokak. In the part of this video, I had explained how Michelson and Stahl in year 1958 experimentally proved that DNA replication was semi-conservative. Now in this video, I shall explain the mechanism involved in DNA replication in prokaryotes. And in part 3, I will explain the DNA replication in eukaryotes. Now, as you know, replication is an autocatalytic function of DNA. This process occurs during the S phase of interphase of the cell cycle. The main purpose of this process is to provide the exact copy of DNA to the daughter cells during growth and reproduction. Now, let us see some of the general features of DNA replication. First, DNA replication is semi-conservative, and this was proved by Michelson and Stahl. It is bidirectional, that is, once the replication begins, it begins in both the direction, in the forward direction and in backward direction. It proceeds from a specific point called origin. Now in prokaryotes, usually they have got a single origin point, but in eukaryotes, because of the linear chromosomes, there are many origin points in eukaryotes. The replication of DNA always occurs in 5' to 3' direction only. And it occurs with high degree of fidelity, that is, accuracy. And this whole process is a multi-enzymatic process. There are large number of enzymes involved in the process of replication. Now let us see some of the important enzymes that are used in DNA replication. The first enzyme is the helicase enzyme. The main action of this enzyme is it helps in the removal of the hydrogen bonds between the opposite strands of DNA and thus cleaving the double-stranded helix into single-stranded. The second enzyme is this DNA gyrase in case of prokaryotes and tropoisomerase in case of eukaryotes. Now this enzyme relieves the strain while double-stranded DNA is being unwound by the helicase enzyme. The third are these SSB protein enzymes. Now this SSB protein enzyme secretes this SSB protein, that is single strand binding proteins, which prevent the rejoining of separated DNA strands during replication. Now the primase enzyme. Now this primase enzyme is responsible for the synthesis of the short primer on the template strand of the DNA. Note, without the primer, DNA replication doesn't start. The next we have this DNA polymerase enzyme. DNA polymerase enzymes are of different types. In prokaryotes, the two most important DNA polymerase enzymes are this DNA polymerase enzyme 1 and DNA polymerase enzyme 3. And in eukaryotes, we have the three most important DNA polymerase enzymes. Those are DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon. Then this DNA ligase enzyme. DNA ligase enzyme, this enzyme is responsible for joining the Okazaki fragments that are formed on the lagging strand of DNA. And also it helps in closing the nicks between the DNA strands during replication and DNA repair mechanism. Now let us see the steps involved in DNA replication in prokaryotes. Now these three main steps are common in both prokaryotes and eukaryotes. The first step is initiation, the second is elongation, and the third is termination. In the initiation process, the origin point is recognized and the helicase enzyme unwinds the DNA helix and the primers are formed. That initiates the replication process. In elongation process, once the primer are established, the new DNA strand is formed in 5' to 3' direction only. So in one strand it will be proceeding in the forward direction and it is a continuous process, and in the opposite strand it will be in the opposite direction, that is in the backward direction, and this strand we call it as a lagging strand where we get this formation of Okazaki fragments. And then after the elongation process is complete, it carries on till it comes to the terminal regions, and after the elongation process reaches the terminal regions, the two DNA strands separate, and this process we call it as termination. So the whole process of replication occurs in three: initiation, elongation, and termination. So now let us see each of these steps in details in prokaryotes. In prokaryotes, at least 10 different enzymes or proteins participate in the initiation process of replication. They open the DNA helix at the origin and establish a pre-priming complex for subsequent reactions. The crucial component in the initiation process is the DNA protein. DNA replication begins from origin. In E. coli, replication origin is called ORC, that is ORC, which contains of 245 base pairs and contains DNA sequences that are highly conserved among bacterial replication origin. Two types of conserved sequences are formed. There are 3 repeats of 13 base pairs and 4 to 5 repeats of 9 base pairs, and these are respectively called as 13 modes and 19 modes. About 20 molecules of DNA protein bind with the 9-mer repeats with ATP, which causes DNA to wrap around the DNA protein, forming the initial complex. The DNA protein and the ATP trigger the opening of the 13-mer repeats, forming the open complex. The two copies of DNA B protein, which we commonly call it as a helicase, binds to the 13-mer repeats. This binding is facilitated by another molecule called DNA C. The DNA B and DNA C interaction causes DNA B, that is the helicase, to open, which binds with each of the DNA strand and helps in the hydrolysis of the hydrogen bonds and brings about the cleaving of DNA strand. So thus the replication fork is formed. The binding of helicase is a key step in replication initiation. DNA B, that is the helicase, migrates along the single-stranded DNA in 5' to 3' direction, causing unwinding of DNA helix. As you can see in this image here, now this is the ORI region, and these are the helicase enzymes. One is moving in the left direction, other one is moving in the right direction. So as I've told you earlier, the replication process is bidirectional, so it moves in both the directions here, and it goes on cleaving the hydrogen bonds between the nitrogen bases here, thus by making the double stranded into a single strand, and this results in formation of a Y-shaped structure, and this Y-shaped structure we call it as a replication fork. The activity of this helicase causes the topological stress to the unwinding strands, forming supercoiled DNA. So this stress is relieved by this DNA topoisomerase or DNA gyrase in case of the prokaryotes by negative supercoiling mechanism. Similarly, the single-stranded binding proteins now get attached to this single-strand DNA so that they prevent the rejoining of these two strands. So that is the main function of this single strand binding proteins. If the single strand binding proteins do not attach to the unwound DNA strand, there is every possibility that these uh, released DNA strands may get rejoined. So hence, to prevent this process, the single-stranded binding proteins get attached to this open strand of DNA and make them single-stranded, and the helicase enzyme proceeds further from 5' to 3' directions. After the attachment of the SSB proteins, the DNA polymerase cannot initiate DNA replication directly. A short segment of RNA nucleotides called RNA primer is formed at the 5' end of each DNA template with the help of primase enzyme before replication begins. It acts as an 5' end of new strand. The primers are then later removed once the replication begins. In case of E. coli, the primer synthesized by primase starts with a triphosphate Ag nucleotide. Primer is always closely associated with the helicase enzyme so that it is positioned to make RNA primer. Once the RNA primers are formed, the initiation process completes, and next the elongation process begins. The elongation phase of replication includes two distinct but related operations. These are leading strand synthesis and lagging strand synthesis. First, let us see about the leading strand synthesis. Leading strand synthesis is more a straightforward process which begins with the synthesis of RNA primer by the primase at replication origin. After the formation of the primer, in presence of enzyme DNA polymerase III, each exposed nucleotide picks up a complementary nucleotide resulting in base pairing. They are picked in the form of deoxyribonucleotide triphosphate, namely D-ATP, D-GTP, D-CTP, and D-TTP. The energy released during this is used in linking the adjacent nucleotide and thus form a new strand of DNA from 5' to 3' direction. Now we shall say about the lagging strand synthesis. The opposite template. Here we call this as a lagging strand because here the synthesis of the new DNA has to wait for some times till some numbers of nucleotides are freed, and then a primer is formed, and then the DNA polymerase III enzyme based upon the complementary pairing will start synthesizing the new strand of DNA from 5' to 3' direction. So each time there is a pause in the replication process, and this pause creates these short fragments of DNA and these short fragments of DNA on this lagging strand, we call them as Okazaki fragments. Now, as and when the replication process proceeds further, the first primer that is formed is removed with the help of the DNA polymerase 1, and then the DNA nucleotides are arranged here based upon the complementary pairing, and then the diester bond between the first Okazaki fragment and the second Okazaki fragment is then joined with the help of this DNA ligase enzyme. So in this way, the elongation process continues both in the leading strand as well as in the lagging strand, and this process continues till they reach the opposite end. Now we shall see the third step, that is the termination step of replication. As the process of elongation proceeds, eventually the two replication forks of the circular E. coli chromosomes meet at a terminus region containing multiple copies of a 20 base pair sequence called TER. The TER sequence are arranged on the chromosomes to create a trap that a replication fork can enter but cannot leave. The TER sequence function as a binding site for the protein TUS, that is terminus utilization substance. The TER-TUS complex can arrest a replication fork from only one direction, so that when either replication fork encounters a functional TUS-TER complex, it halts and the other fork also halts, and thus the replication of DNA gets completed. Replication of DNA separation opposing replication forks leave the completed chromosomes joined as catenanes or topologically interlinked circles. The circles are not covalently linked, but because they are interwound and each is covalently closed, they are then separated by the action of topoisomerase. In E. coli, a type 3 topoisomerase known as DNA topoisomerase 4 plays the primary role in the separation of the catenated chromosomes. It breaks both the strands of one chromosome and allows the other chromosome to pass through the break, and thus helps in separation of this catenated chromosomes, and thus the two daughter chromosomes separate from each other, and thus the termination process ends. Now, this is a summary of the DNA replication. First, the ORI regions are recognized by the helicase enzyme. At the ORI region, the hydrogen bonds between the nitrogen bases are cleaved, and thus the DNA helix becomes unwound. Then after the DNA helix is unwound, the single-stranded binding protein stabilizes the unwound parental DNA. Then the DNA polymerase enzyme synthesizes continuously a new strand of DNA from 5' to 3' direction. The strand on which the DNA is synthesized continuously, we call this as the leading strand, and the opposite strand where the DNA fragments are synthesized, that is the DNA is synthesized in short fragments, these we call it as the lagging strand, and these fragments we call them as Okazaki fragments. These Okazaki fragments are again rejoined with the help of the DNA polymerase 1 enzyme, and ligase DNA. And thus, once the elongation process ends, they come to the terminal end. Once they come to the terminal end, with the help of this DNA topoisomerase 4, the two DNA chromosomes are separated, and thus the replication process ends in case of prokaryotes. So, here we conclude this topic on the mechanism of replication in prokaryotes. So in part 3, I'll tell you about the mechanism of replication in eukaryotes.